Yeah, I had a feeling. But.
All right, so this is already on? Microphone's already on.
This one's on.
Microphone's already on? All right, great. So we have people on Zoom watching as well. I want to welcome everyone to Matt Elliott's defense. I want to say a couple of words before we get started. Remind everyone that we do have people on Zoom as well as here in person. When this is all done, we will have a series of questions where we'll deal with everyone in the room first, and then we'll deal with the questions on Zoom, and then the committee, which is myself, Scott Hamilton, and Matt Bracken, who's on Zoom, will go in the other room and finish up the formalities before we come out and do a little bit of fun stuff. So really excited for this. It came on, I don't want to say like surprise, but relatively rapidly. I was informed that I needed to be in this room at a particular time by Matthew and Scott Hamilton as well. Just realized it was in person because Matt decided that there was a timeline that had to happen, and we all made it work. Matt came to us in fall of 2017. Matt came to us in the fall of 2017. From the great state of Minnesota, the land of 10,000 lakes, famous for Prince, right?
Yes.
The Mall of America, but not seaweed biologist. He's a very humble kid. He came and said he was actually surprised that I accepted him to the lab because he had no business doing seaweed work. He had no background, nothing. And I didn't believe him. There was something about him there was something about his application when I first met him that indicated that regardless of the seaweed side that I thought Matt was going to do well at Moss Landing. And then one of the most famous phycologists ever, Josephine Tilden, came out of Minnesota. And I thought, you know what, you can be a phycologist out of Minnesota. Matt had potential. He did great here at Moss. His classes were great. He did well in biology at seaweeds, marine ecology, which I think you later TA'd for Scott and I during COVID, if I remember correctly.
Yeah.
He got the Simpkins Award and the Myers Award as well while he was here. Matt was always extremely helpful and reliable in the lab and in the field, and he ended up as one of our trainees, along with his buddy Max, who's here, on our first real aquaculture grant, no impediments grant that Scott Hamilton and I did. Matt learned a lot about abalone, seaweeds, abalone and seaweeds together in integrated multi-trophic aquaculture, and then quite a bit about seaweed nutrient dynamics, which ultimately led to his thesis, which we're going to hear about today. Along the way, he became one of the most critical employees at our seaweed farm, Monterey Bay Seaweeds, where he helped develop many of our seaweed culture techniques that we currently employ. And this ultimately landed him a job as an aquaculture technician at Hub SeaWorld Research Institute in San Diego, which he got 2 years ago. So it's been 2 years that Matt's been gainfully employed in the aquaculture industry, traveling more than I do currently, giving talks all over the place, which is great, even before he graduated. So I just wanted to say before we fully introduce him, that's a testament to his perseverance and his integrity that he came back. He had to get through COVID. He had some personal hardships, and then he went off and got a job and has a great life and decided that it was really important to come back and finish. And he has a fantastic thesis that he wants to show you today. So with that, I'm going to introduce Matt Elliott and spatial and temporal variability in nutrient physiology of Pyropia Perforata.
Great. Thank you for that introduction, Mike. Yeah, let's jump right into it. It's going to be a little long, so buckle up. And just for reference, this is a photo of some baby Pyropia Perforata to start this presentation. So algae play a critical role in marine ecosystems due to their ability to uptake compounds such as carbon and nitrogen from the ocean. Nitrogen is often referred to as the most important compound for algae, as it is the most limiting factor for growth. Atmospheric nitrogen enters the ocean in gaseous form and is transformed through the nitrogen cycle into usable forms like ammonium and nitrate. These usable forms of nitrogen are referred to as nutrients and play key roles in manipulating a seaweed's nutrient physiology. Nutrient physiology in seaweeds studies how nutrients are acquired and used for growth, health, and energy. In seaweeds, this often involves measuring nutrient uptake, growth rate, and nutrient storage. When nutrients are first uptaken from the ocean, They are absorbed through either passive transport, which doesn't require energy, or active transport, which uses energy from the sun. Once absorbed, nutrients are either stored or used for growth. The acquisition and utilization strategies of nutrients in seaweed are properties that make each seaweed's nutrient physiology unique and potentially fluid. Nutrient uptake is an important parameter with regards to seaweed, as it can explain changes in physiology and changes in the environment. Nutrient uptake rates are excellent indicators of a seaweed health and growth strategies. These rates are often described by the McKillis-Menten function, which calculates uptake rate based on maximal uptake rate, nutrient concentration, and half the saturation value. Furthermore, the graph on the right shows the relationship between substrate concentration and uptake rate, highlighting three phases: externally controlled uptake at low concentrations, internally controlled uptake at intermediate concentrations, and surge uptake at high concentrations. To draw comparisons, uptake rate can be used to understand changes in environmental conditions, like upwelling or the effectiveness a seaweed can have on nutrient removal, such as bioremediation. While nutrient uptake rates are important, nutrient storage plays a critical role in seaweed survivorship. Seaweeds, specifically those in the intertidal zones, can adapt by increasing their storage capacities. Critical nutrient concentrations, which measure the percentage of a nutrient element in seaweed tissue, indicates the nutrients available for growth. For instance, Macrocystis pyrifera can use its nitrogen reserves for up to 3 weeks without negative growth impacts during low nutrient periods. This ability to store and use nutrients is common in many seaweed species, allowing them to survive prolonged nutrient scarcity. On the flip side, when nutrients are abundant, seaweeds can engage in luxury uptake, storing excess nitrogen for times when nutrients are scarce. This stored nitrogen enhances their nutritional value, as higher nitrogen content often correlates with increased amino acids and proteins. Red seaweeds, in particular, have higher nitrogen content because of their abundant phycobility proteins, making them more nutritious and preferred by consumers over brown and green seaweeds. The study by Olson in 2020 highlights that red seaweeds generally have a higher protein content, which is reflective of their higher nitrogen content, and makes reds a more valuable nutritional resource compared to green and brown seaweeds. Furthermore, impacts on nutrient physiology can occur as a result of environmental heterogeneity, where metrics such as irradiance and abundant nutrient levels significantly impact seaweed physiology. These factors vary spatially and temporally due to seasonality, geography, and upwelling. For example, upwelling in California is directly impacted by wind. Stronger and more consistent upwelling favorable winds in Northern California result in higher upwelling intensity compared to Southern California. Upwelling brings nutrient-rich cold water to the surface, providing high nitrate concentrations, while ammonium levels fluctuate more due to rapid uptake by other autotrophs. This regional difference significantly influences the nutrient availability in these areas, affecting the growth and nutrient physiology of seaweeds. Understanding these environmental factors is essential for studying seaweed, which can be a key diet for many marine species and humans. Changes in environmental heterogeneity could result in changes to the nutritional value of a seaweed species like Pyropia perforata. Pyropia perforata, commonly known as nori, is a highly valued in aquaculture for its broad distribution and high internal nitrogen content, ranging from 3.5 to 6% compared to 2 to 2.5% in giant kelp. Furthermore, by multiplying the percentage of tissue nitrogen by 5, we can estimate the protein content. Moreover, because of Pyropia's extensive geographic range, shown by the figure on the right, where you can see it ranges all the way from Southern California up to Alaska, and the nutrient timing that it has as a wild species, it is an excellent species for understanding nutrient physiology and understanding how it adapts to varying environmental conditions. The study aims to understand how the percentage of tissue nitrogen and total protein concentration in Pyropia perforata varies geographically and seasonally. The California coast presents significant variability in environmental factors affecting nutrient concentrations. However, the relationship between ambient nutrient concentrations and seaweed nutrient physiology remains poorly understood, highlighting the potential variability in nutritional value of seaweed. And then additionally, from here until the end of the talk, I will refer to tissue nitrogen and total protein concentration as percent N and TP, or total protein, but interchangeably. So building on our understanding of the environmental heterogeneity and its impact on Pyropia perforata, I first hypothesized that percent N and total protein in Pyropia perforata would be significantly higher during high-intensity upwelling periods and in northern locations. Secondly, I hypothesized that nutrient concentrations was significantly positively correlated with percent N and total protein. And lastly, that abiotic factors would significantly modify this relationship between percent N and total protein. And then this is one of my collection sites. This is Gaviota State Beach, or pretty close to Santa Barbara area. The first objective was to assess the spatio-temporal variability in percent N and protein concentration in Pyropia perforata along a latitudinal gradient. This involved collecting samples from various sites, cleaning them, and freezing them for later processing. I collected Pyropia perforata from 5 locations across California, spanning 4 degrees latitude to capture the environmental heterogeneity. This map shows the collection sites from southern to northern California. Additionally, Moss Landing Marine Labs is shown on the map to indicate the central location where all samples were transported to. Samples were collected from February to November, and months were grouped into seasons, such that March, April, May were the spring, summer was June, July, August, September, October, November was the fall, and December, January, and February was the winter season. The Pyropia samples were collected monthly at each site during the Pyropia perforata season using a 100-meter transect, and then the samples were transported back to Moss Landing Marine Labs, where they were cleaned of marine debris, rinsed with DI water, dried with paper towels, and then frozen for CHN and protein analysis. The second objective was to determine if ambient nutrient concentrations correlated with percent N and total protein in Pyropia perforata. Samples were collected solely from Faneuil Beach, then transported back to Moss Landing Marine Labs, and then cultured at specific nutrient concentrations. To test this, live Pyropia perforata was cultured in 100-gallon tanks for 10 days at a stocking biomass of 100 grams, with pH maintained through daily PCO2 additions. Pyropia was weighed using a net drying method established by a previous pilot study that compared 3 different stocking biomasses to 3 different wet weight drying methods, as shown by the graph. A coefficient of variation determined that 400-gram dried using the 5-minute method had the lowest amount of wet weight variability. Moreover, Pyropia was pruned to maintain daily nutrient concentrations and uptake consistencies. Pyropia was cultivated with specific nitrogen species, either nitrate or ammonium, and specific concentrations over all trials, ensuring consistent nutrient additions and controlled environmental conditions. The tanks were maintained at ambient seawater using heat exchangers, and nutrient concentrations of 0, 5, 15, and 50 micromole were used to replicate natural fluctuations. Water quality was monitored using YSI and hobo loggers to measure parameters like temperature, salinity, pH, and dissolved oxygen, ensuring stable conditions throughout the trials. And then after a trial concluded, Pyropia was cleaned, weighed, and frozen for CHN and protein analysis, just like Objective 1. And then lastly, the third objective was to determine the correlation between percent N, total protein, and abiotic factors. CHN and protein results from the previous objectives were used to answer this question, as well as data from Senkus. Frozen samples were dried at 60 degrees Celsius for 48 hours, then broken down into a powder using a ball mill, in order to make the samples homogenized. You can see the image on the left is an image of some dried pyropia. The image in the center is the ball mill. And then the image on your far right is an image of a processed prepped sample. In order to measure total protein in Pyropia perforata, I used a 3-phase extraction method at Moss Landing Marine Labs, which involved methanol, chloroform, and millipore H2O. This process started by weighing dried, homogenized Pyropia perforata to 25 milligram and placing it into Eppendorf tubes. Methanol was added, vortexed, and centrifuged, followed by chloroform and millipore H2O, with each step involving vortexing and centrifuging to separate the layers. The chloroform layer was sonicated to ensure complete separation. The interface layer was dried and rehydrated using a urea buffer, sonicated again, and then vortexed and centrifuged. This created multiple aqueous layers, which were stored at 4 degrees Celsius until the protein assay was used. And then these layers were all used to calculate total protein for each sample. The Pierce BCA protein assay kit was used to determine the total protein concentration. This method followed the Thomson 2008 protocol, which involved using 2 reagents that created a colorimetric value based on the levels of amino acid present in the sample. The colorimetric value was then run through a spectrophotometer to obtain accurate levels of absorbance, and then this method was compared to the traditional percent nitrogen multiplication method to test its accuracy and reliability for Pyropia perforata. And then CHN was used to measure precise measurements of percent nitrogen gas. This involved weighing 1 to 3 milligrams of homogenized Pyropia perforata powder and placing it into little tin capsules. And then the image on the left and the right show the process that was used for prepping samples for CHN, where on the left you see taking a little spatula and placing some of the Pyropia sample into the tin capsule, and then the image on the right is using a microbalance to measure accurate weights. And then these samples were sent to UC Santa Cruz for CHN analysis. To complement the percent N and protein concentration data, real-time data loggers from Senkus were used to obtain a range of environmental data, including sea surface temperature, chlorophyll A, wind speed, photoperiod, solar radiation, as well as 2 different upwelling indices, coastal upwelling transport index and biological effective upwelling transport index, referred to as QT and BUT. Additionally, sea surface nitrogen concentrations were estimated from each site using a regression model equation from Ghost et al. 2000 that uses sea surface temperature and chlorophyll A to estimate nitrogen concentration. Data analysis included linear mixed models for spatial temporal variability and COVAs for nutrient correlation and multiple regressions for the relationship of percent N, total protein, and abiotic factors. And then this is an image of one of my treatments where you have the 4 different nutrient concentrations, and this was with ammonium. So for objective 1, I wanted to see how does Pyropia perforata percent nitrogen and protein concentration vary in time and space. So I began collecting Pyropia perforata in February 2021. Before February 2021 or February, none of the sites had harvestable size Pyropia. So this was established as the start of the Pyropia season as seen on the table. And then the last collection month varied by each site, with the southernmost site concluding their seasons first in August and September, followed by the northern sites in October and November. So therefore, the length of Pyropia season and number of seasonal replicates varied between sites. So you can see that Bodega had the longest collection period of 10 months, while Gaviota had the shortest period of collection with only 7 months, since the Gaviota season ended in August because it wasn't available to collect in September. It had disappeared. All right, so the results showed that the interaction of season and site did not have a significant effect on percent N. However, seasonality did have a significant impact on percent N, with the highest levels observed in the early Pyropia season, as shown here on the graph, where spring and winter tend to have a higher percent average N compared to summer and fall. And then additionally, site did have a significant impact on percent N, with percent N highest at Bodega Bay during the winter and spring, and percent N lowest at Cayucas during the fall. And then additionally, you can see the season reps between each site and how those vary between each season, and how the winter section, there aren't error bars because there's only one replicate for each site for that winter season. Moving on to total protein. Season and site did not show a significant interaction effect on total protein. However, there was a significant difference among seasons, with the highest total protein in spring and winter and the lowest in the fall, as seen on the graph. Additionally, Fanshell Beach had the highest average total protein, while Cayucas had the lowest average protein. However, site did not have a significant effect on total protein, further emphasizing the seasonal variability in protein content. The graph on the left shows a significant positive association between percent N and latitude. The northernmost site had the highest percent N. Even though the southernmost site did not have the lowest percent N, there was still a significant association. However, total protein did not show a similar trend. There is no association between total protein and latitude, and we see that from the p-value and the R-squared. So for objective 2, further wanted to see how ambient nutrient concentration related to percent N and total protein concentration in Pyropia perforata. So I collected Pyropia perforata from Fanshell Beach starting in February, like the objective 1 trial, and then this went through October 2021. Through this time period, I conducted 13 trials in total. 7 trials used nitrate as the primary nutrient, while the other 6 used ammonium. And then, like objective 1, the trials concluded when Pyropia perforata disappeared from Fanshell Beach. So nutrient uptake of tumble-cultured Pyropia perforata showed significant depletion after 4 hours, particularly in higher nutrient concentrations, which is shown on the graph on the left, where 50 micromole had the most depletion from 50 to 15, while the 5 micromole treatment only went from 5 to maybe 3 for nitrate and maybe 2 to 1 for ammonium. The biomass-specific uptake rate was highest at 50 micromole and lowest at 5 micromole. And you can see this on the graph on the right, where the x-axis is the nutrient concentrations and the y-axis is the uptake rate. But moreover, the nutrient depletion was consistent across both nitrate and ammonium cultures. Pyropia perforata cultured at 50 micromole had the highest percent N for both nitrate and ammonium cultures, with the lowest percent N observed at 0 micromole. And this is further shown on the graph on the left, where the x-axis is the set nitrogen concentration and the y-axis is the percent N. Moreover, there was a significant interaction effect between nutrient species and nutrient concentration on percent N. Additionally, nutrient species and nitrogen concentration had a significant effect on percent N, with ammonium cultured Pyropia perforata having a higher average percent nitrogen compared to those cultured in nitrate. And this is seen on the graph on the right, where your x-axis is the nutrient species and the y-axis is your percent N. Furthermore, the results indicated that percent N increases with higher nutrient concentration, and the slope of percent N versus nitrogen concentration changed as a result of nutrient species. Total protein was also highest at 50 micromole for both nitrate and ammonium cultures, and then this is further seen on the graph on the left. Similar with the previous 2 graphs, where the x-axis is the nitrate concentration and the y-axis is total protein. Although there was no significant interaction effect between nutrient species and nutrient concentration on total protein, both nitrogen concentration and nutrient species individually significantly affected total protein. Pyropia perforata cultured in ammonium had a higher average total protein compared to Pyropia cultured in nitrate, and then this is seen on the graph on the right. So the overhanging results of this was that protein levels increased with higher nutrient concentrations, and that nutrient species also had an effect on on total protein. Differences in pigmentation were observed in Pyropia perforata when cultured at different nutrient concentrations. Low nutrient concentrations appeared lighter in color, and this is seen in image A for both nitrate cultured and ammonium cultured, and then they appeared darker, which is seen in image D for both nitrate cultured and ammonium cultured. So to test the third objective, I wanted to assess how abiotic factors correlated with % N and protein concentration in Pyropia perforata. So I considered factors such as temperature, solar radiation, day length, chlorophyll A, wind speed, the two upwelling indices of QD and beauty, and estimated nitrate. There was a significant correlation between % N and total protein in Pyropia perforata. Samples with the higher % N consistently had a higher total protein level. The ratio of total protein to % N was 4.9, which is very close to the 5 factor from Engel et al. 2016, and this was a good confirming factor for the methodologies used. Multiple regression analysis revealed that environmental parameters significantly predicted % N in Pyropia perforata, and this is shown from the model statistic at the bottom. Solar radiation and day length both had a negative association with % N, and then the graph on the left shows solar radiation versus % N, and then the graph on the right shows day length versus % N. Additionally, chlorophyll A was negatively associated with % N, while estimated nitrate concentration was positively associated with % N. The other environmental parameters of temperature, wind speed, the upwelling indices did not have a significant association with % N. For total protein, the multiple regression analysis found that environmental parameters were not significant predictors of total protein. However, temperature showed a significant negative association, indicating that higher temperatures were linked to lower protein levels. And then this is further shown by the graph where temperature is your x-axis and total protein is the y-axis. And then you can ignore the D on the graph, that shouldn't be there. And then moving on into the discussion, this is an image of some cultured Pyropia perforata. I know you've seen some images of Pyropia from this presentation. This one was unique. You can see that the pigmentation is almost red. Oftentimes, Pyropia kind of looks like much darker in pigment, almost like brown to black in color. So the changes in pigmentation were something that was a big discussion point from the results of this thesis. Nutrient physiology in seaweeds, including % N and total protein, was closely linked to various abiotic factors such as seasonality and geography. Pyropia perforata, with its broad geographical range and intertidal ranges, served as a great model species to study these influences. The study aimed to understand how seasonality, geography, and abiotic factors, which included sea surface temperature, solar radiation, wind speed, chlorophyll A, day length, and estimated nitrate, as well as the upwelling indices, QD and beauty, affected nutrient physiology in Pyropia perforata. I found that seasonality had a significant effect with the highest % N in total protein in the early season. Geography had a significant impact on % N, and abiotic factors showed to be a significant predictor of % N. Lab experiments further revealed that nitrogen concentration positively affects % N in both ammonium and nitrate cultures. However, nitrogen concentration did not significantly influence total protein. Pyropia perforata cultured in ammonium had higher % N and total protein compared to those cultured in nitrate. This indicated that spatial and temporal variabilities significantly affect percent N, with ammonium having the biggest impact. Seasonality played a significant role in affecting both percent N and total protein in Pyropia perforata. I observed that these values were lowest in the fall and highest in the lowest in the fall and highest in the winter-spring seasons. This can be attributed to the life cycle of Pyropia perforata, where early life stages show rapid nutrient uptake and storage. Additionally, the dynamic California current ecosystem, characterized by significant seasonal changes in nutrient availability, also influences nutrient physiology. These seasonal patterns highlight the importance of considering temporal variability in nutrient studies. Seaweeds in the early parts of their life cycle are adapted to uptaking nutrients more rapidly, therefore moving nutrients into storage and to build up nitrogen reserves, also to facilitate health and growth in the seaweed. In the fall, upwelling intensity is low due to offshore relaxed wind intensity, therefore having a reduced amount of nutrient-rich seawater interacting with intertidal seaweeds. These stages in the later part of their life cycle explain why their nutrient physiology is influenced by seasonal and geographic variations in nutrients and in other environmental conditions. Seasonality played a significant role in affecting both percent N and total protein in Pyropia perforata. I observed that these values were lowest in the fall and highest in other seasons. Oh, whoops. Sorry about that. So this study further found that geography significantly impacted percent N, with the highest values at the northernmost site, Bodega, and the lowest at the southernmost site, Cayucas. However, geography did not have a significant impact on total protein. The geographic scale of the collection and the environmental heterogeneity between sites could have influenced these results, highlighting the need for more extensive sampling to understand these patterns better. This underscores the complexity of nutrient dynamics across different geographic regions and the necessity for localized studies. Environmental heterogeneity influences such as nutrient concentration, solar radiation, and temperature would change as a result of latitude. However, it's important to note that the southernmost site, Gaviota, is south of Point Conception. Point Conception is a well-known biogeographic boundary for many marine species because it is where the Northern California current meets the Southern California current. North of Point Conception, the coastline aligns with northwesterly winds, increasing upwelling intensity and nutrient availability. South of Point Conception, the coastline curves eastward, reducing upwelling intensity and nutrient availability. This geographic difference in upwelling intensity and nutrient availability highlights the importance of considering local environmental conditions when studying nutrient physiology in seaweeds. There was a significant positive correlation between nitrogen concentration and percent N, but not with total protein. This indicates that while higher nutrient availability leads to increased percent nitrogen, it does not necessarily translate to higher protein levels. Seaweeds like Pyropia perforata can retain nutrients for extended periods of time without becoming nutrient limited, as demonstrated by my findings. Even at zero nitrogen concentration, Pyropia perforata maintained higher than expected percent nitrogen levels, suggesting that a 10-day trial may not be sufficient to fully deplete nutrient reserves. These results align with findings by Hanasek in 1993 and Gerard in 1992 on nutrient retention in seaweeds. Additionally, this could be due to the presence of non-nitrogenous protein material in seaweed tissues, which can affect the relationship between nitrogen concentration and total protein. Understanding these dynamics is crucial for accurately estimating protein content in seaweeds. Similar observations have also been made in other studies, such as those by Lorenko et al. In 1998 and Naldi and Wheeler in 1999. Furthermore, these findings highlight the complexity of nutrient metabolism in seaweeds. Pyropia perforata cultured in ammonium had a higher percent nitrogen value compared to those cultured in nitrate. This suggests differences in how these nutrient species are assimilated and stored by the seaweed. Ammonium is rapidly metabolized into amino acids, while nitrate can be stored in higher concentrations within cells. These findings highlight the importance of nutrient species in influencing nutrient accumulation and metabolism in Pyropia Perforata. Research by Pedersen and Borum in 1997 and Bracken et al. In 2006 show the notion that seaweeds exhibit preferences for nutrient species based off their physiological capabilities and life histories. And then the graph on the right is from Bracken and Stachowitz in 2006, showing how different species have a different preference for uptake of nutrient species. While comparing percent N to total protein to see if they could be proxies for each other, given that total protein is often estimated by multiplying nitrogen by a factor of 5, my study found that the Pierce PCA protein assay provided a conversion factor close to this study. And the relationship between total protein was linear. This suggests that percent N can be a reliable proxy for total protein in Pyropia Perforata, although some variability exists due to method differences. This is consistent with the findings by Engel et al. 2016. Understanding this relationship is essential for accurate nutrient analysis in ecological and in aquaculture studies. There were noticeable differences in pigment color between nitrogen cultures, as seen by the images on the screen. Pyropia Perforata grown in low nitrogen cultures showed a lighter color, while those in high nitrogen cultures exhibited a deeper red color. This is consistent with findings that higher nitrogen levels enhance phycocyrene and phycocyanin, the main color determinants in Pyropia. Gao et al. 2019 demonstrated similar results in a different Pyropia species, where higher nitrate and PCO2 levels resulted in deeper pigment colors. These findings suggest that pigment color can be an indicator of nutrient availability and health in seaweeds, providing a visual cue for assessing nutrient status in natural and cultured populations. Additionally, environmental factors significantly impacted nutrient physiology in Pyropia Perforata. There was an inverse correlation between sea surface temperature in both percent N and total protein, indicating higher values in cooler water. Solar radiation, chlorophyll A, and day length were significant predictors of percent N, suggesting that lower radiation and shorter days are more favorable for nutrient accumulation. However, there was not a significant relationship between environmental parameters and total protein, further highlighting the complexity of factors influencing photosynthesis in seaweeds. And then my findings from the study also align with other studies such as Phillips and Hurd 2003 and Britton et al. 2020, emphasizing the need for a holistic approach to understand nutrient dynamics in a marine ecosystem. To conclude, nutrient physiology in seaweeds is fluid and influenced by various factors. My study found that seasonality significantly affects percent N and total protein, with the highest values observed in the northernmost sites. Laboratory trials showed strong positive correlations between nutrient concentrations and internal nitrogen, and estimated sea surface nitrogen was positively correlated with percent N. These findings underscore the importance of considering local environmental conditions when studying nutrient dynamics in a marine ecosystem. They also highlight the need for a more comprehensive approach to understanding nutrient metabolism in seaweeds, integrating both field observations and controlled experiments. Future research should aim to include a wider geographical range to capture the full extent of environmental heterogeneity influencing nutrient physiology in seaweeds. Additionally, there is a need to focus on the role of non-nitrogenous protein in seaweeds, which can provide deeper insights to nutrient metabolism. Investigating the link between environmental parameters and nutrient physiology, as well as the relationship between nitrogen and protein, will help us better understand how seaweeds respond to varying environmental conditions. And then to start the acknowledgements, first I'd like to thank my advisor, Mike Graham. He mentioned this initially, but you know, I had no right being here. There's a lot of, there's a, you know, I was 22 when I moved out here. I applied for grad school. My summer going into my senior year of college, and by some sheer luck, I got in. I don't know, maybe Mike liked that I played soccer or something, but, you know, he gave me a chance, he gave me an opportunity. He let me learn about seaweed, tumbling it, and, you know, the field that was available and the opportunities that could come from it. So, forever indebted. Thank you so much. And then Scott Hamilton, he put me on my first grant, my first project, which was on IMTA, or Integrated Multi-Trophic Aquaculture, which is probably why I have my job now at Hubs, which I know they're listening, so they're probably cracking up. But yeah, like learning about dynamics between seaweed and invertebrates, even though you're a fish guy, immensely helpful for my career. And you know, both of you have had such a strong impact on my career and like where I can go in the future, and you know, things I didn't really think were feasible for me a few years ago, or even in Minnesota. There's no oceans there. There's no tides. So, and then lastly, Matt Bracken, I want to thank you for being on my committee. I know I first met you, it must have been 2018, when you came and did a seminar here at Moss, and we shared a couple of sakis at a sushi bar. No, your insight about nutrient uptake has been so helpful, and you're obviously at the top of your field when it comes to this kind of thing in intertidal community structures. So all of your support and comments on my thesis from then up until this point have been very helpful as well. And then some more acknowledgments. I know people tend to have very illustrious slides. I put this together a little bit faster than that. But yeah, I want to thank the beer pig community. It's an ever-growing community of phycologists trying to learn something. So a lot of names up here and a lot of names I missed. But you know, every Tuesday when we had our meetings, it was always insightful having an older student with a beard just kind of chop you down when you're standing up and trying to talk. And you know, those, it might seem funny, but you know, those types of reality checks when you talk about research do play a part, especially in your career, because you need to come ready and come prepared and be ready for someone to say something that maybe isn't nice. But you know, beer pigs, we had a lot of fun. I learned a ton, so forever grateful. The Shore Lab, Mike alluded to it. I worked at a seaweed farm, but there's other stuff that happened at the Shore Lab, like the IMTA project that I worked on with Max and Scott. But yeah, no, like definitely my aquaculture knowledge stemmed from the Shore Lab, so very happy for that opportunity. I don't see the shop guys here today, but I know James and Billy were very helpful in my path at Moss Landing, whether it was working on my own personal stuff or working at stuff at the Shore Lab. They were very helpful. And then other MLML staff, Tara does so much for students, and then other people from MLML staff that helped me along my way. And then, of course, I couldn't have done this without my financial support from the Myers Fund and the MLML Simpkins Family Marine Science Scholarships. And then I didn't add this in here, but the Monterey-based Seaweeds emblem definitely helped pay some bills too. So, very grateful for that. And then lastly, I have a little collage. I'm horrible at taking photos. This is like every photo in my phone, honestly, from Moss Landing. So it was good I was here for 7 years, because this is 7 years. But yeah, you know, my sister flew in from Portland. So happy she could be here. Truly the most important family member I have. And then Max is here. We met in grad school, I think on our first day. We've been pretty good friends ever since. Yeah, and then I know Dan's listening on the Zoom. Dan was immensely helpful for my thesis. Hoping to see him soon. Steve's in a tank at the bottom. Steve was super helpful as well. And then, of course, Sean. You know, we met right outside those doors on the first day of orientation, and you know, we've been inseparable ever since, and I'm lucky to call her my partner. I know there's so much more to say, but yeah, I think I'll leave it at that. But with that, yeah, I'll take any questions.
Well done. So, like I said earlier, we're going to have some questions here, and then if we can get any on Zoom, can you see Zoom there? And do you have the Zoom up here, or is it inside the room? I'm trying to figure out who would be able to communicate. If not, not Zoom. I mean, yeah, Zoom.
I don't think I do it from here. I think
Okay.
I do it over there.
So, Matt Hess, text the beer pigs and ask if anyone on. Can anyone join the Zoom from their phone just to see if there's any comments coming through? I'm going to bring a prop while we do that. So we're going to have questions, and I just wanted to kind of highlight, what's going to happen afterwards. There's a little bit of food when we're done. There's a little bit of beer. There's a little bit of seaweed partaking, which we'll leave for later. I just want to point this one out. This was actually made by Matt's former employer at Monterey Bay Seaweeds, Erica Graham, and just to honor him, it is a homemade cupcake topped with sugared and candied nori, which, as you now know, counterbalances the 35% total protein. So this is like the epitome. Well, I guess it's not really keto. Maybe if she didn't coat it with sugar, it would have been a little bit more keto. But 35% protein is pretty high for putting into a cupcake. So, it's going to be that much more when you guys get to enjoy them. So I am going to take questions. I have two, and I'm going to ask one now, and I'm going to ask one when it's all over with. Matt knows this very well, and the beer pigs know this very well. I, until very recently, was adamant that you are not allowed to devise a thesis in the phycology lab focused on anything applied. So as you know, there's a lot of aquaculture related to Matt's thesis, but he had to come up with this, his whole thesis design and presentation really without saying the word aquaculture. You didn't hear it very much today. So he justified it on the ecological importance of what was going on. I think he did a fantastic job doing so. So my first question is on the ecology, and now I want to dig to see how deep that ecological knowledge really is. So I think it was like essentially your first data graph. You just blew right past it, where you showed the month at which every one of your local sites first had nori arrive. And it was always, anyone remember? February. Your sister answered. Look how I'm, so February, right? No, not this one. I want the one, right? There you go. Look at the month that it ended. So I need you to explain. This has nothing to do with nitrogen as it relates to your thesis. It might have nitrogen in the answer. I want you to answer for us why this conspicuous and, I don't want to say charismatic, but very common species in the intertidal, regardless of geography, shows up in February, but then is geographically dependent on when it disappears.
That's a great question.
I know. I've been
So-
waiting for 7 years to ask him that question.
I think it's driven by a couple factors. One would be recruitment. But then, you're talking about a species that at the end of its life cycle, it's sending all of its spores into the water. So then where recruitments are going to land is where the water's pushing. If you argue that the spores are always going to go where the winds are pushing more coastline, then it's always going to go to a more upwelling hotspot, which would explain why it has this maybe not so much geographical trend, but Bodega, the highest upwelling site from my study, had the longest season.
Okay, so now I'm going to dig a little deeper. Why is it always February when you first see it, regardless of where you are?
I mean, this was 2021, so maybe it changes with respect to time and place. But why is February for all the sites? The life cycle of Pyropia is photoperiod dependent. So as the photoperiod increases, that allows these gametophytes to become sporophytes or become adults. So.
Okay, so now I'm going to go deeper on the last one.
You
said they fell apart. No, the last part, the last month collected. You said they fell apart, and therefore you couldn't collect anymore. So they fell apart. They died in August, they died in September, they died in October, they died in November. From your field experience of seeing all these populations die, how did they pass away, and is there any indication why they were? Because I'm trying to understand why they die earlier in the year in the south and later in the year in the north, beyond your dispersal argument.
So I guess I would argue that the southernmost sites, not only is your seawater warmer, but your light intensity is higher too. So as you're senescing, if you will, you're really pushing all of your nutrient reserves into growth and health. So at some point, you're going to run out. So whereas in your northernmost sites, those impacts are lesser. So you're able to extend your life cycle, if you will.
Right on. So the recruitment's photoperiod dependent, which has nothing to do with geography, and the other one is local environmentally dependent.
Yes.
Cool. All right, good answer. Anyone else before I hand it to Scott? Because you know what happens when I hand the microphone to Scott. And it'll all come back to pee, right? Somehow it's going to be fish.
So when we were working with the monkey-faced eels in aquaculture, we found that, they love to eat this species like crazy. It was like their favorite thing to eat. So if I wanted to feed a bunch of monkey-faced eels and get them to grow as fast as possible, what conditions should I grow your Pyropia in to make it the best, most nutritious seaweed, in Mike's seaweed farm? What should I do to grow that seaweed to feed to these monkey-faced eels?
Yeah, it's a good question. Clearly, like the season of Pyropia is long, and then it goes through a lot of different types of environmental changes, whether it's like how intense the sun is or how much nutrients you're being fed. But I think if we were to go back to this graph here, there seems to be a trend with upwelling. So I would argue that you want a higher nutrient concentration when you're feeding your seaweed in order to get the most nitrogen or most protein. But then additionally, you have to be cognizant of getting too much light. So in the summertime, if we're eclipsing photoperiods of 14, 15 hours, it's probably too much, because then it's growing too much. Because if you feed something nutrients, it's going to grow unless you take away one of the other factors for growth, which is light. So if you minimize the light a little bit but still feed it as much nutrients as possible, then you're likely able to maximize those nitrogen reserves, which could internally turn into protein.
And feed them more fish pee.
Fish pee did great. I didn't forget about fish pee. Clearly, the orange line is ammonium, and then orange is ammonium on the other graph too. Percentin was higher in ammonium, and is a better, higher concentrations. Ammonium metabolizes so much faster.
I'm getting visions of like the movie The Martian with him feeding his own potatoes. So that's great. I did want to point out that your uptake rates were similar. So even though uptake of ammonium and nitrate were relatively similar, what you're saying is assimilation and packaging into proteins is really what drives it.
Yeah, and there's another study I didn't mention here too. Is I think it was Thomas, 1998, that essentially stated that seaweed species like Pyropia perforata have a nitrate holding period, where when they take up nitrate, it takes a while before they can actually use it. And sometimes that's often seen in uptake rate, where it's the first 15 minutes of uptake, you won't see it in the tissues, and it takes time for it to translocate nitrate throughout the cell. So.
Well, I'm checking. It doesn't look like we have any questions on Zoom. Uh-oh. Do you have a direct?
Talking about this slide, I see higher nutrient uptake rate at higher concentrations. Does that result in more biomass of the individual, or did you see anything like that? Because I think you said that it was only a slightly higher increase in percent nitrate and not much change in total protein, but that's a lot more nutrient getting uptake in, so I'm just wondering what the plant is doing with all of that.
Yeah, and it's doing a lot of things. When seaweed takes up nutrients, it doesn't all just go right to growth, right? It can. It's possible if you manipulate the right abiotic factors. If you give it a lot of light and a lot of surface time with the light, then it's going to want to photosynthesize rather quickly. But if it's still uptaking nutrients, it's going to do one or two things. It's either going to hold on to the nutrient, or it's going to use it right away for growth. Those strategies are complex. You can kind of emphasize the strategies by changing abiotic factors, but why they do it at what specific times, you would kind of need a little bit more elaborate study looking at exact photoperiods and maybe a little bit more deeper dive at like what's happening when nitrogen goes into the tissue. But it seems like it's like a middle ground, whereas uptake rate might not necessarily change with respect to abiotic factors, but what happens with those nutrients will change, and that's just a matter of abiotic factors. No.
Hey Matt, how do you think your findings with this intertidal nori species would compare to the nitrogen physiology of subtidal nori species also present in the California current system?
So this is funny because Dan's thesis, he focused on Pyropia nereocystis, which is an epiphyte of bull kelp. They're not comparable, right? You're talking about an epiphyte that is on a host of a kelp species that doesn't grow subtidally by itself. However, but if he was referring to, let's say that there is a Pyropia species that grew subtidally versus an intertidal species that goes through the fluctuations, I would argue that the differences in nutrient physiology of a subtidal species are less likely to variability to the same extent of an intertidal species because of the lack of not being submerged, the solar radiation that they're getting from the sun and just being fully exposed to it without any like shading effect from being subtidal, and then the adaptation strategies of an intertidal species where they go through periods of not being submerged under seawater, so they've had to adapt to hold on to nitrogen at higher concentrations in order to survive these fluctuations. So I would argue that it's less variability.
So a kid from Minnesota does understand a bit about tides. All right, any more? Okay, so I'm going to ask my last question, and it's selfish because I want to know the answer. You showed lots of variability in geography, and I think it was a really nice, clear pattern of going from south to north. It increased %N, it increased total protein. There were a lot of reasons why. And you learned much of that by doing the experiment you did at Moss Landing, which was only with fan shell, which was right in the middle. How might you, and we were kind of looking at maximum in the geography of about 35% total protein.
Yeah.
When you did the fan shell and you did the experiment, you got around that, around 30. What would it have looked like if you had used bodega that habitually has higher protein? Because, as you said, it has higher nitrogen for being an upwelling zone. Had you used that in the experiment, do you think you would have pushed the upper boundaries a bit of how much protein loading you could do, or do you think they ultimately do cap out?
I don't think site-specific Pyropia would have changed if I'm always culturing it in Moss Landing. If I changed the culture site to maybe bodega and I took, a seaweed species that wasn't at bodega, I think I could get similar results to bodega Pyropia because it's matching that environmental homogeneity. But like
I'm gonna put a little bit more oomph into this. I'm going to invest $100,000 in a land-based nori farm. One's going to be in Bodega, one's going to be in Monterey, or Moss Landing, let's just say. Let's just look at the Moss Landing one. Where do I collect my nori for that farm? Right? Because now my money's on the line. Do I go get it from my local environment, like Fanshell, or do I send somebody up Bodega to continue to bring that down so that I can farm it at Moss Landing? Right now you're suggesting there would be no difference and that you could take it from either.
Yeah, I would say take it from the local source. I think the manipulations that happen when you culture a seaweed species are indicative of the abiotic factors that you are conditioning the seaweed to.
All right, did that jog any, let me make sure it's not Matt Bracken texting that he's actually got a question. Yes, he does. Holy moly. I'm just going to ask one of his questions, and we'll ask the other one in committee. Okay? Well, let's just say it. So I want to. We'll demand a question. I want to know why %N responds to nitrate, whereas total protein responds to temperature. And I guess what he's asking is there something about protein that responds more to temperature specifically?
Yeah, it certainly could be that temperature maybe forces nutrients to metabolize faster. So maybe that's why you see that curve a little bit stronger, especially when you get to the higher temperatures. you really see that drop off in total protein. And then what was the other part of his question about like the nitrate side?
That was it. I mean, the point of it is that percent N responds to nitrate. That was one of the nitrate concentration, whereas total protein was responding to temperature.
Okay. Yeah, so this is much more.
Right?
Yeah. Yeah, this is much more positive association. So when exposed to nitrogen, seaweed can take it. And it's shown very clearly that if you give it more, it's going to hold more. But then as soon as that nutrient goes into the seaweed, the patterns and metabolisms that occur may not be directly associated with protein. But temperature is something that can drive metabolism, can drive photosynthesis, may have that stronger effect on moving protein or utilizing whatever reserves are being used for amino acids and driving them to something else.
All right, thank you. We're going to cut them loose. Thanks, everyone on Zoom and YouTube for watching. Thanks for all of you for being here. Thanks, Matt and his family for making it here and doing it in person. I know it's an effort, and it was fun. We're going to steal him for a very short period of time. The rest of you can hang out. There's a keg of beer. There's cupcakes. There's pizza. Please make sure that there is at least a cup of beer, a cupcake, and a piece of pizza for Matt when he comes out, and that you haven't all eaten them. And for Scott, so 2. I already had my cupcakes, and as you all saw on the video, I had my beer as well. But with that, one more big round of applause for Matt. Really proud that he made it back down here and finished strong. So congratulations, and let's go ahead and start.